Yes. Welcome all guys for the today's webinar, that is animal toxicity studies by Dr. Karthik. You all may be knowing, many have you all have attended in AI in pharmacology, also artificial intelligence in pharmacology, which was taken by Dr. Karthik. Hope this will also be very good session by him. Yeah, over to you, Karthik.
Am I audible?
Yeah, you're audible.
Yeah. Thank you, Dr. Raghu. I again want to say thanks to Samruddhi Elite Educators for giving me this opportunity. And today I'm going to talk about animal toxicity studies. So can I share the screen?
Yeah, should we wait for another 2-3 minutes, or shall we start, Dr. Karthik?
Okay, fine. We will start at 4:05.
Okay. Is the audio clear? Please put yes in the chat box. Dr. Karthik, you share your screen.
Yeah, okay. Should I share the entire screen on window?
Yeah, entire screen.
Entire screen, okay.
Yeah, and open the PPT.
Okay, I just shared the screen. Is it visible?
It is visible. Yes, guys, give a thumbs up if the screen is visible. If any query, you can message in the chat box. Okay, guys. And Trushti Ratwa, you can message in the chat box. Okay, Dr. Karthik, I think we can start. Can we start, guys? Okay. Dr. Karthik, that below there is one tab, like stop sharing. You can hide it. You can hide it. Yes.
Yeah, I did it.
Okay. Yes, once again, welcome you all guys to the Samruddhi Elite Educators webinar on animal toxicity studies. Yes, today Dr. Karthik will be dealing with animal toxicity studies. Any issues, you can message in the chat box. Okay. Yes, over to you, Dr. Karthik. You can start.
Yeah, thank you so much. Good afternoon, everyone. I will be dealing with animal toxicity studies. So this is one of the most important topics in our experimental pharmacology, which everybody should know as a postgraduate, as an MD pharmacologist, or other pharmacology respected educations as well as professionals. So this one right now, there are so many things which are coming up, and firstly I'll be going with some basic things, and then I'll be dealing with all the other recent things which are happening right now in the industry as well as in the real time, and with some examples. So basically I'll be dealing with These contents, that is introduction, necessity of doing animal toxicity studies, then CCSCA guidelines, the classification of the toxicity studies, then the governing bodies which gives proper guidelines, and the types of toxicity tests done, and some alternate animal models, and the recent trends, and I'll be telling the summary and the gist about all the things what I spoke about. So coming to the introduction, so basically animal toxicity is what deals with something that is toxicology, that is nothing but study of poisons, and which are concerned with the study of adverse effects of xenobiotics or biotransformation. So dealing with all the PK parameters, pharmacokinetic parameters like absorption, distribution, metabolism, and excretion, and some other pharmacodynamic related things like dosing intervals, dose frequencies. Everything will be handled by taking these toxicity studies which are done in the preclinical phase, where we find out the exact dose for the human population, that is the maximum tolerated dose for the humans. So Casseret in 1996 defined it as a science that defines the limit of safety of chemical agents for human as well as animal population. So just a quote of the day where Paracelsus just give a quote telling that poison is in everything, and no thing is without a poison. So the dosage makes it either a poison or a remedy. So this quote was done. This quote was given during 1896 by Paracelsus, and then things started to happen about people started thinking about toxicology studies, then chemical toxicity, then environmental toxic particles which were studied during that time. So this was a very good quote given by him. So why do we need to do these animal toxicity studies? That is a necessity. So first one is development of new drugs and extension of therapeutic potential of existing molecules. So mainly to develop new drugs and to for a better therapeutic outcome for a particular diseased condition. And coming to the next one, that is risk benefit ratio can be calculated. So to know how much benefit it is going to be given to the population, human population, and how it is going to help us. So that is why we do this animal toxicity studies, to determine that safety parameter, and to predict the therapeutic index as well as a therapeutic window. So we know therapeutic index and therapeutic window, which is the safe range which can be given. That is the range where the therapeutic outcome is better and the adverse effects are reduced and the therapeutic outcome is maximum. So that can be found out with the help of animal toxicity studies. So it is actually given in animal toxicity, it is given as TA, that is therapeutic index, that is maximum tolerated dose by minimum curative dose. So that is the formula which is the most important formula which is used. And so when the ratio of this therapeutic index is small, that means there is better safety of a drug or a particular chemical entity or a new drug moiety, or it can be any other chemical substance. So that can be taken care of, and toxicology studies can be done. And to even know the adverse effects, and to how much amount of adverse effects is it going to cause in the long run or in the short term, and how it is going to affect in the future of a particular patient. If we give a particular drug or a particular medicine to a human for a particular patient or a particular person, so we get to know what is the exact thing, how much is the required amount of drug to be given. So that is the basis of knowing why we should do this animal toxicity studies. So coming to this, previously these are some of the governing bodies where the guidelines are given for the particular animal studies. So first one is the basically mostly all of you will be knowing, that is CPCSCA, that is Committee for the Purpose of Control and Supervision of Experiments on Animals. So recently that has been changed to CCSCA, that is Committee for the Purpose is removed from the CPCSCA, and now it is termed as CCSCA. That is nothing but a Committee for the Control and Supervision of Experiments on Animals. And whatever research proposals, whatever the research study or you're going to do, it has to go through this IAEC, that is Institutional Animal Ethics Committee, that has to approve your study. Then only we can go for the study proper, so that we can conduct a study. And to do that study, we need to have this some important standards, that is good laboratory practice, that is GLP, that has to be certified. For a lab to function for animal toxicity studies, we should have this good laboratory practice where it has to be taken care of, and all the guidelines are taken care of, so that everything, the environment, the place where we do the animal experiments, the area of the place, the cleanliness, the sterility, everything has to be taken care of in that. So those are those are some of the guidelines which they give, and it has to be certified. Then only an institute or a pharmaceutical industry can conduct, or a pharmacy institute can conduct this particular animal toxicity studies. And coming to this OECD, that is Organization of Economic Cooperation and Development. So this body also tells about different tests which are done in animal toxicity studies, where they validate the tests and they tell that whether this test is sensitive or specific or not. So basically that is what OECD guidelines do. And also they also help in, it's almost like an integration actually. So they try to integrate with IAEC, CCSA guidelines and good laboratory practice. And so this is a particular chain of governing bodies where we require particular things which are required to conduct an animal toxicity studies. Okay. So before going to the topic proper, I want to say these are some of the experimental animals which are used. So it is classified under rodents, non-rodents, and miscellaneous group of animals. So in rodents, we have rat, mouse, guinea pig, and hamster. And coming to non-rodents, you have rabbit, monkey, dog, cat, and pig. And some miscellaneous like frog, pigeon, zebrafish, chicken, and a Drosophila. So now I told you, right? So CPC-SCA guidelines have been changed into CCSCA guidelines. The only difference in this is just the term. Otherwise, most of the things are similar only, as similar to CPC-SCA. But why they did that was to improvise their own guidelines. And now you can even go to their website also and check. Now it is termed as CCSCA guidelines only. So they're trying to tell that prevention, than doing for a prevention, we can just control or check for the proper supervision of experiment on animals. So what is basically the goal of these guidelines were to help in giving a good proper guideline for handling the animals, taking care of the animals while during experiments, and for the well-being of the animals. And that should be respected, because we are doing experiments on animals, so that animals has to be respected, and they have their own stand. So that is what is important, the goal of doing giving these guidelines for a particular institute or a industry to conduct an experiment. So basically in these guidelines we have, the first one is your veterinary care. So you should have a proper veterinary person or a particular physician or a particular qualified veterinary person to take care of the animal house. So every institute which are having this IAEC as well as the nomination rate, so that will have a separate animal house in their institute to conduct an experiment. So that is what the veterinary care tells about. The second one is your animal procurement. So you have to, the transportation of animals. Normally, if the animals are not available in your institute, when you try to procure it from someone else or from the government or from other institutes, the proper guidelines of procuring it and with the transportation, the safety of the animals has to be taken care of. And quarantine and stabilization. So once you procure the animals, so you need to quarantine the animals. So for a small animals like mice, rats, you can quarantine for up to 1 week. But for bigger animals, you need to have the good amount of time, that is around 6 weeks. Like pigeon, like pig, mouse, all the big animals has to be given at least 6 weeks of quarantine, because they would have stayed or lived in a separate environment. They could have had a disease in them that should not be transmitted for the existing animals which are already present in the institute when you procure it, and so that is why we need to cage them and quarantine them, and we need to stabilize them, and they should also get adjusted to the temperatures, the environment over that new area, right? So that is why this quarantine is very, very important. And coming to the next one, that is after quarantine, you need to have a thorough checkup of the animal, whether it is having any disease. So if there is any disease, you have to diagnose it, you have to treat it, and you need to manage the disease. And then only it is fit for the animal experiment. And definitely animal care and technical personnel should be appointed for the animal house throughout 24 hours. The person has to be there, a technical person, to take care of the animals, and proper instructions has to be given to the person, so that he takes care of the animals, so that there is no unwanted deaths or unwanted diseases which will get contracted in the animal house. And the personal hygiene. So once the technical personnel, he has to have his own personal hygiene, like wearing gloves, having some sterile washes, or whatever kind of sterile environment he has to have for a particular technical personnel who handles the animals. So even the veterinary physician, anybody, so whoever is there in the animal who takes care of the animals has to have a proper personal hygiene. And also animals hygiene also has to be taken care of. And now recently in this guidelines, they have told, previously in CPCSCA guidelines, this restrainer use was not having that much proper guidelines. They were just telling that the restrainers can be used, but now the CCSCA guidelines and amendment of CPCSCA has told that restrainment, that is, restrainers are nothing but they are the ones which try to hold animals, or you try to stabilize the animal. They are the restrainers. Like there are so many types. Either you can put it to the neck, either you can put it to the hands of the animals, or the limbs, or any other parts of the body, so that it has a proper stability for immobilizing the animal. So now they say that the restrainers has to be used temporarily, only to only if you are conducting any experiment on it or not. Otherwise, restrainers should not be used for managing the animals or holding the animals. So that is a very strict guideline which has come out for this. And definitely the corridors, the floors, the drains, and the storage areas. So all this in the animal house has to be totally taken care of. The corridors has to be clean with sterile environment, and it has to be regularly cleaned. Then the floors, then the drains. Normally the drains are not that much important, but if you are having a proper cold storage or closed storage places, there the drains has to be very clear. So where the proper draining of other debris or water or cleaned things has to be thrown out, or it should be drained properly. And for storage areas, like see now, if we can have a cold storage area as well as normal storage area. Normal storage area can be some items or some ingredients which has to be kept there. Feeding substances, all the things has to be stored there. And if you have a animal tissue, or if you have a debris, or if you have a dead animal that has to be stored as a cadaver, so you need to have a proper cold storage room also. So basically these are some of the CCSA guidelines for any animal house which has to maintain, or which they have to take care of. So coming to the types of toxicity studies, so you have so many toxicity studies. Like first one is a systemic toxicity studies. In that you have something called a single dose study, that is acute toxicity studies, and repeated dose the studies, that is subacute and chronic toxicity studies. So you have reproductive toxicity studies, then you have local toxicity studies, then you have hypersensitivity tests, then you have genotoxicity tests, then you have carcinogenicity, then you have mutagenicity tests, embryo toxicity tests, and neurotoxicity tests. So I'll be dealing with I'll be giving a gist of all the tests. So, and I made sure I don't bore you guys, so I made it very diagrammatical, as well as more less of words, just going to explain with only images. and make sure you understand little bit at least. Just for the initial stages, I'm going to talk about some texts which are there, but other things I have good amount of images which I'll be showing you guys. So coming to the first one, that is very, very important, that is your acute toxicity test. So in this test, that is your systemic toxicity test. So it evaluates the adverse effects that occur following exposure of organisms to a single or multiple doses of a test substance. So this is within 24 hours by a known route, that is oral, dermal or inhalational route can be done. So what is the basic funda of doing this is to find out the LD50, that is the lethal dose of that is where there is 50% of death in the test population. So that has to be calculated. So that is the most important thing about doing acute toxicity test. So where the drug is exposed for a short-term period. And they are the only standardized toxicity tests where death is the intended endpoint. So here is the only test where we have the death, that is your intended endpoint. So here in this test, you need to have as I mentioned in the classification, the rodents and the non-rodents. So you should have at least a rodent and a non-rodent to do this test, which is very, very important point. And you should have at least 2 routes of administration, that is one which will be proposed for the humans. So either you can have oral, inhalational, either intravenous or other what do you say, the parenteral routes can be used, intraperitoneal, intrathecal, everything can be used. So at least 2 routes has to be taken care of. But now if we want to use oral administration for humans, that is the first route which has to be given to the animals. So different dose levels has to be done for 14 days. So this study will take care of at least 14 days. So this is the minimum period which has to be done. That is your acute toxicity test. And one more type, that is your repeated dose toxicity studies. So it actually comes under acute toxicity test only. But here in this, the difference in that is you have, instead of 14 days, it will be given for 28 days and then evaluated. So it is between 14 to 28 days you need to give the dose in different strengths. So this is the person, that is J.W. Trivan. He is the one who proposed the use of 50% lethal dose, that is LD50. So that was the major breakthrough in animal toxicity studies to know what exactly is the proper effect of chemicals or drugs in the body. So this he determined the lethal dose for individual chemicals. So he is the one who was behind the toxicity tests, and he was the one who tried to improvise acute toxicity studies. So as I told, here in acute toxicity studies, you have something called as LD50. So this was a test which was done by Trevan in 1927 for testing substances intended for human use, such as digitalis, that is digoxin, and insulin. So that was the first one which he did to find out the LD50 values. So it covers the application of one high dose or several low doses during 24 hours. So here you can see it should be given, the several low doses has to be given with one high dose for 24 hours, and the effects are observed for 14 days. So that is the basic thing about this acute toxicity test, and the endpoint is the death of animals. So here in this image, what I've shown you here, a mice is given some random 2000 mg, that is 2 grams per kg extract. Orally, single limit dose has been given, and it has been checked for 14 days. This is the high dose which is given, but normally they will give for serial doses, that is at least for 5 or 6 serial low doses will be given, 2 grams, 1 grams, then 500 mg, 400 mg, 300 mg, 200 mg, 100 mg, like that. So that will be given for a period of 24 hours to check the lethality. So wherever there is at least 50% of the test population will be checked. So if the 50%, if now we take 6 animals, so in that 3 animals are dead at 1 gram per kg extract. That means that will be given, that will be our LD50 value. So that dose will be taken as a LD50 value. So, and it has to be observed for at least 14 days. So here, now after giving this extract or a drug or a chemical, they have checked heart muscle damages, then liver toxicity, then renal toxicity, and some blood dyscrasias. Like all the enzymes are checked in the liver, then heart, you will be checking some infarction, ischemia, all the damages which will occur in the heart. All that can be checked by after doing the autopsy, after the death. So if death occurs, we can go for an autopsy and we can check what exactly are the problems or the histopathological changes which has occurred due to the chemical entity. So this is your acute toxicity test. So before doing anything, CCSA guidelines have told that there is for animal research you need to follow this 4 hours. Before it was only 3 hours, now it has become 4 hours. Now even they are telling it is also 5 hours, but will be more concentrating towards 4 hours because that is there in the literature. So 4 hours are the most important hours which has to be followed. That is reduction, refinement, replacement, and responsibility. So reduction is what means why they have done this is because to reduce the toxicity to animals because of the chemicals. So reduction is either you try to reduce the number of animals to a minimal so that the information does not hamper, but still we get a good information about the drug. And refinement is try to improvise the study parameters or statistical tests and other parameters, materials and methodology of the study can be refined so that we get a better outcome for a particular study. And replacement, so instead of taking higher animals, we can go for lower animals and where alternative techniques can be done, which I'll be dealing later. And the last one which was added, that is your responsibility or something called as rehabilitation. Okay, so this is the aftercare. Now see, now we have done, now I just told you rat has been given 2 grams of an extract or a drug. So that after the experiment is done, and if the rat survives, so for the survived rats we have to take proper care. The rehabilitation after the experiment has to be taken care of. That will be missed by almost all the experimental physicians or pharmacologists or any research scientists or anybody who's into the animal experiments. So that brought out CCSA to tell this IEC committees, which told that this one more thing which has to be put in the guidelines, that is your responsibility or rehabilitation. That is very important for animal ethics. Yeah, so here for finding out some LD50 values, you have different types of methods. So first one is your FDP method. So FDP is nothing but fixed dose procedure method. And next one is your ATC, that is acute toxic class studies, and UDP, that is up and down procedures, and some other article which is there. But these three are very important, where you can see all are introduced during 1990s. And you can see the accuracy levels of all the three tests are good. So in this first test, that is fixed dose procedure, that is FDP, is what is important. That is because you will be giving proper fixed dose to a particular animal at intervals of time during 24 hours. And coming to this one, normal ATC is nothing but it is a conventional method only. But the only difference in this is you'll not be given lot of several extra doses. And one more thing about this is number of animals are very less in this. That is only 3 to 12. And UDP method is up and down procedure, where you give one high dose and one low dose. Again, you give back the higher dose and the low dose. That is how the things happen. That is how the procedure chronologically it will happen. So these are basically the three types of toxicity tests where we try to determine LD50. So what important exactly in this table you need to know is the first three things which are important, and the cost. Cost has been reduced a lot. Previously, the methods which were Trevan was using was very costly, but these tests have improved the top cost and economic feasibility. So it's a very good things, and it's still running and it is still done. Then the duration also has reduced because of this introduction of all these tests. Then reproducibility is very good, and the endpoints. So you need to see the endpoints. So here in FDP, you are just seeing the signs of toxicity, but in others, you're seeing like ATC and UDP, you're seeing all the things like signs of toxicity as well as the death. And obviously, all the three require regulatory approval from the IAC committee, and it has to be properly given. The data, the methodology has to be given how it is done. And these are some of the in vitro tests and in silico tests, which are in acute toxicity tests, which determine LD50. So these are some of the tests which are used. So here you can see in the first three, that is 3T, NRU, cytotoxicity test, NHK, and 3T3, neutral red uptake, that is NRU, phototoxicity test, and cytotoxicity tests. So they require regulatory approvals. So, and this is your in vitro methods. And in silico methods, you have neuroblastoma, SEMA cell line approach for botulinum neurotoxins in acute toxicity. Where for these three tests, you don't require any regulatory approvals, but still the test or the procedure has to be done in a very meticulous manner. Coming to the next one, that is your subchronic toxicity. So you have either same in this also, you need to take a rodent as well as a non-rodent. And here, instead of 14 days, we give a drug for at least 90 days. Okay. And then what we do is we try to do weekly body weight variations are checked. Monthly biochemical and cardiovascular parameters are observed. Then gross and histopathology is observed. And intra-individual variations should be less, and weight variation should be less than 20%. So this is very, very important. This last point, that is intra-individual variations of animals should be very less, and weight variation also should be less than 20% difference between the animals. So normally in a test group, we take at least 6 animals. Then even it can go up to 12, 18, 24, we can take like that. But minimum test group animals has to be taken is in a group is around 6 animals. So we should have all the proper parameters to be taken care of, and the body weight has to be monitored. The monthly biochemical parameters has to be taken care of. Cardiovascular parameters has to be taken care of. And later, when there is a death during the period of 90 days, we need to do the autopsy of the animal and try to remove all the organs, vital organs, and try to find out try to get the gross and the histopathology, and give it to a pathologist to know what exactly has happened, or what is the report of a particular drug or an entity which has caused any issues in the organ system. And whatever dose is taken from for this subchronic toxicity is from your acute toxicity studies. So whatever study, acute toxicity study, whatever dose you have got, from that you can do subchronic toxicity. The extension of this is your chronic toxicity. Same here also, rodents and non-rodents are taken, and the test drug has to be given for more than 90 days at least. And this chronic tox, as the name says itself, chronic means it's a long-term effect. So the long-term effect can be studied, and it can be extrapolated to human safety studies. So that is why chronic toxicity studies are done. Mainly where chronic toxicity studies are done is in your cancer studies, your immunological studies, then which requires a chronic like hypertension, diabetes, all that can be done with chronic toxicity. And essential for new drug entities also. So because why it is important is because new drug, we don't know the safety parameter properly. On a long-term efficacy or safety, we don't know. So for that also, we have to do this chronic toxicity test. So that is very important for chronic toxicity. So here, just a small pictorial representation of a particular extract, that is Cistus ladaniferus, where acute toxicity studies and subchronic toxicity studies were done. And you can see here, after doing for 14 days of acute toxicity and 90 days for subchronic toxicity, the toxicity profile, the LD50 is more than 5 grams per kg. So they would have started at least from 2 gram per kg, 3 gram per kg, or a high dose, that is your 10 gram per kg can be given for the particular animal. Then they would have got for 7 grams, 5 grams, 4 grams, 3 grams. So there they got to know that in acute toxicity, the LD50 value is more than 5 gram per kg here. and NOAFL, that is your no observed adverse event levels, that is NOAEL actually, where there is no adverse effects which are observed. That is in the dose range of 1 gram per kg. So at 1 gram per kg, the adverse effects were minimum. So what we got to know, the LD50 we got to know, the maximum therapeutic effect we got to know, and we got to know the adverse effect profile with the help of acute toxicity as well as subchronic toxicity. And see, as you can see, they have taken the plasma, they've plotted a graph here, and they have taken this histopathological studies of different times, from the days, weeks, months, everything which is taken care of, and then the study has done. So this is how normally a study goes on for a particular toxicity studies. So to know the adverse effects of a particular drug entity. Yeah, this next one is your mutagenicity. So mutagenicity is nothing but any mutations which will cause some growth of an organism or something, mainly because of any mutations. So normally genetic, other non-genetic, polygenetic mutations, then different types of mutations. So that can also be checked with the help of this test called as Ames test or Amy's test, where the rat liver extract is taken for Salmonella strain. This requires histidine. So they have taken two plates here, one with the possible mutagen that which causes mutagenicity, and the plate which is normal as a control. So when this has been added to the media with minimal histidine, so you can see after incubation, the control plate has very less growth of or any other pathological parameters are less. But compared to this the next one, that is your uh, test plate, where we have high number of revertants, histidine minus and histidine plus, suggests that mutagen can cause mutation. So this proves that this mutagen which is there in this Salmonella strain can cause some pathological issues, can cause mutations in the genes. So that is the test where we check for a particular drug to know whether a drug is having any anti-cancer property or a drug which has cancer property. So recently we had this thing right about ranitidine. So it had its own cancer potential. How they got to know is by trying to do all these kind of tests to know whether it is having some cancer potential or not, or any other mutations. Not just cancer, it can be anything: immunological disorders, diabetes, hypertension, then normal or infections, everything. So that all can be checked, and the treatment can be given, and then it can be checked whether this treatment is going to help or not. And coming to the next one, that is your carcinogenicity. So you can see again the animal liver extract is taken. Before that, we give the chemical treatment, the drug treatment is given, and then the liver is removed, or the liver extract is taken out, and then they do something called as the gene expression analysis with the help of these microarray technologies, and the whole genome is being checked. And this Tox Plus is what is a software or a chip which has toxicity profiles which are already embedded in the data, and then the computer tells, after feeding the data, the computer tells that whether this has a particular carcinogen or not, the chemical has some carcinogen property or not. So this, for that you have something called as carcino screen. So that is a very old software which was done with the help of this gene expression analysis. So here you can see there is a graph here, there is a carcinogenic potency. So when the data is given an input, so here you can see if the graphs are in this side, that is the red lines, red graphs, bar graphs. So that shows that it has some positive carcinogenic property, and the green one tells that it does not have any carcinogenic property. So this basically predicts the carcinogenicity of a particular chemical entity. And the next one is your genotoxicity studies. So here in genotoxicity studies, we try to take rat or a mice, and then they take this extract from the liver, that is your S9 extract from the liver, and then they give something called as strain of S. typhimurium of both sides, A1 and A2, and then they go, before that they do incubation, and then they give a test compound to these particular strains after extracting the liver extract. Then later it has been put inside the agar, and the test compound, and then it is incubated for at least 2 to 3 days. So there is two things, that is revertant colonies and colonies. Revertant are nothing but group of organisms which are grown. So revertant means which is exacerbate or exuberant colonies which are produced. As you can see in this controls, there are only two things are there, two growth of um, animals or organisms which are there in the plate. But here revertant colonies of organisms which is there because of the particular genotoxicity of a particular test compound. Now F type S typhimurium, the amount of organisms are more here because the test compound has lot of genotoxicity. So that is why it tells about the genotoxicity of a particular test compound that can be also be evaluated. And the next one is your reproductive toxicity. That is recently it has come up, and it's doing it's very helpful actually. And this one where we try to do this embryo toxicity studies, reproductive toxicity studies with the male as well as the female rats, mice, any other animals, rodents and non-rodents, where the pregnancy is induced, and then with the help of this gestation days, as you can see in this right diagram where I have put this one, the black and white diagram, where the gestational days are 0 to 6, there is nothing has to be done. But after the 6th day, we give a test compound to the animal, and 4 groups of 24 mated females are checked in this particular experiment. And then at the time of 17th day and the 20th day, we try to induce abortion, and we try to remove the fetus during 17th to 20th day, and then the fetal examinations are done for to know if the test drug is having any teratogenic property, or it is having some other reproductive issues, developmental anomalies, then reproductive organs, the sexual maturity, which I have depicted in your left hand side. That is the period of age which is taken care of, from the period of fertility till your sexual maturity. So the above is your humans, the down part is your animals, where they try to find out. So with the help of this studies, to know, mainly to know if a drug is having any teratogenic property or not. And yeah, this one, the next one is a very important test, that is your neurotoxicity tests, where lot of recent developments have come. And this also, here also, embryo development stage only we can do this neurotoxicity studies from the before the birth of an animal, or mainly for the tests which are done, right? So for that, from the embryo level only we can find out if a drug is having any neurotoxic potential or not, because we have a lot of neurological disorders which are having less number of treatments, and if we have even treatments, it is not sufficient. So like Alzheimer's, dementia, all of them are like potential areas of research, and it is being done extensively, and these neurotoxicity studies are done in a very extravagant manner all over the world, all over the globe. So basically the brain development, the cell-based assay, and the zebrafish assay. So this one is there, the last one, that is your zebrafish. So that was a that was a recent one which came up, and it was very useful test for finding out the neurotoxicity for in vitro methods, which was one of the alternative animal model to find out neurotoxicity, and it has got very good results, and a lot of research is happening on zebrafish. And so many conferences recently they are having hands-on workshop on zebrafish to know the pentylene tetrazole, that is your convulsion property, to know the anti-convulsion property, then for dementia, the Alzheimer's, the antioxidants which are required to improve the brain architecture. So all that tests are done with the help of this zebrafish assay. As I told you, the neural development, the network formation, beautifully. You can see these beautiful images. It is small, but but really good images which tells about the brain architecture. If it is not visible, I'll just try to expand it, but I guess it is visible. All the MRI images, the contrast images, fluorescent images, which tell about different types, the network formation, how the synaptic connections are happening in the brain, then behavioral testing, how the behavior of the animal is changing, or particular how it is going to affect the human beings. So all that is being studied with the help of zebrafish. And the next one is your local toxicity studies. You have local toxicity studies like dermal toxicity studies, ocular toxicity, vaginal toxicity, rectal toxicity, and inhalational toxicity. So these are some of your local toxicity, which is not systemic. So mainly it is topical, or it is having mucous membrane penetrable properties like vaginal, rectal, all that are having mucous membranes which have a lot of which will overcome first pass metabolism, all those things where the maximum tolerated dose is checked. So these are some of the animals which are used, like guinea pigs, rabbit, and rats. Dogs are also used. And to evaluate, they have used erythema, then redness in the eyes, the dermal. I'll just show you with the images in the next slide. Then the vaginal toxicity and the rectal toxicity. So in the vaginal toxicity, the swelling as well as the closure of introitus or the vaginal opening can be checked. And rectal toxicity, the signs of pain, blood, mucus in feces, condition of the anal region or sphincter. Then inhalational toxicity to find out the respiratory rate, the histological examination of respiratory passages, all that are being checked. So here you can see inhalational, dermal, vaginal, and ocular. These are some of the examples which are done. So as you can see here, no, the inhalation, the animal is kept in a cage where the aerosols are given, and then the lungs are, the lung parameters are checked with the help of respiratory rate, and other pulmonary function tests are all checked. All the drugs which are given inhalational route. Dermally, skin patch test or any infiltration of drugs can be checked, where you can see there is some dermal toxicity here in the middle image, where the test drug has caused this dermal toxicity. So we can know the hypersensitivity reactions which has occurred. And your vaginal toxicity studies. You also have in vitro as well as in vivo tests. So in vivo, as you can see here, the vaginal opening, the test drug is given, and then they check whether there is closure of the introitus, how is the mucous membrane of the introitus of the vagina, to know whether it is having some any vaginal toxicity or not. Because so many drugs which are given intravaginally, the pessaries and other contraceptive methods. And the ocular, which is the very famous experiment which are done on rabbits, where it is still going on. And nowadays the simulators have come. Now the animals and rabbits are reduced it is very less actually. And this is what is about all the local toxicity studies. Now you can see in this, right, all the 4 images, you can see there is so much harm to the animal. There is so much toxicity for the animals. The health of the animal is totally jeopardized. So you can see ocular, you see the eyes of the rabbit, it is red and it is having some ulcers. Then the skin of the rabbits, all that has been very badly affected. So that is what the importance of my topic today is, to know what can we do other than doing all these tests which are already there. Now what are the recent things which have come for the next future trends, where the trends are changing right now. Now the amount of animal toxicity studies which are done is very less, and lot of other techniques have come which are almost equivalent to in vivo techniques. And definitely all the tests which are done to evaluate quantitatively, we have different types of instruments like UV visible spectrometry, infrared spectroscopy, gas chromatography, and HPLC, that is your high performance liquid chromatography. So all this will give you a better picture of the values of toxicity parameters. Then ADME predictions can be done, that is your pharmacokinetic predictions, that is absorption, distribution, metabolism, and excretion. All that parameters quantitatively can be assessed with the help of these instruments. There are many other techniques also, but these are some of the basic things which you should know. And nowadays, other than HPLC, we have UPLC, that is ultra high performing liquid chromatography, where it gives proper plasma concentrations, and it is very, very sensitive. The test is very sensitive, and we get a very good accurate results. So all this, what I showed you here, this showed that so much of injury is happening to the animals. So we need to have some alternate methods for this. So firstly, I want to tell you guys about the benefits of this alternate to animal models. So one thing about it is, first one is animal-free testing may be more accurate. So sometimes animals testing, the whatever the results we got, it might not be correct for the human population. So animal-free testing can be done, and non-animal testing may be more accurate than tests done on animals. So that's what I told. So it might be more accurate, or it might be less accurate also. Then alternative testing saves the lives of animals. So the lethal dose, the amount of lethality, the morbidity, mortality of animals are reduced when alternate to animal models are used. Then alternative testing may be faster, definitely, than the conventional methods. The recent techniques which have come, it has speeded up the amount of time, reduce the amount of time for a particular test to happen, for a particular result to get to obtain. Then non-animal tests may be cost effective. So definitely it might be cost effective also. And alternative to animal testings are more environmentally friendly. So these are the so many benefits are there for doing alternate to animal models. So definitely there are, as I told you in this 4 Rs, the second one, that is your replacement. So replacement of animals from higher animals to lower animals. So these are some of the animals which are taken as alternate animals, animal model or mammalian models to do the tests. So in this we have chick embryo, zebrafish, brine shrimp, uh, copepods, Daphnia magna, mollusca, roundworms, and rotifer. So in this, no, right now not all animals are used. Mollusca, chick embryo, zebrafish are the most extensively used. Zebrafish, chick embryo, and mollusca, that is snails. These are some of the animals which are used right now, and other animals are also used. Alternate animals models, mammalian models are used. Yeah. So what exactly have we replaced from animal tests? So these are some of the tests which are replaced from animal tests. That is your cell-based tests. That is your in vitro tests. In silico tests like QSAR, that is qualitative structure activity relationship assay. Then tissue engineering methods, molecular dynamics, and molecular docking. So these are some of the animal tests which are replaced with the help of in vitro techniques and in silico techniques. So yeah, so here we can see some in vitro models and in vivo models where we have cell culture methods, then fruit fly, that is also called as Drosophila melanogaster. So very, very important, and this one also is very, very important alternate animal model which is used. And as I told, zebrafish. So cell culture, you don't have to know about the advantages and the limitations, but for Drosophila and zebrafish, you should know why are we taking these animals. So basically, Drosophila, it is easy to work with. It has a short generation time, and it has a low cost of maintenance. Okay. And zebrafish, the advantages are it has high reproductive rate, development is external, then genetic similarity to humans are more. So when we take a one zebrafish or two zebrafish, the reproductive rate is very, very high. The amount of zebrafish you can reproduce, it can be produced even to millions also, hundreds, thousands to millions also. So that is a very good animal which is used very much commonly for experimental animals, and Drosophila also. So definitely one, if an animal has a advantage, definitely it will also have a disadvantage also. So these are some of the limitations which are there, but I won't be going detail about this. And see here you can see the zebrafish. So many types can be, so many tests can be done. That is a behavioral testing. then pharmacological aspects, then oxidative stress, new tools, then locomotive tracking, then toxicological studies are done, then neurotransmitter studies are done. So many applications are there for this zebrafish. So yeah, this zebrafish, as I mentioned, it's a very important animal which has to be known, which is the recent trending topic. So where the you can see the life cycle of the animal. So within no time, you can see from 0 hours to 2 days, 3 days, 4 days, you already have a zebrafish. Within the fourth day, you have a from the ova oocyst or the ova, you already have the zebrafish. That is your that is what the amount of reproductive rate. So within 4 days, you can produce a zebrafish, right? So you can produce so many within few days of time. Where you try to do this something called as bioimaging, then neurotoxicity studies, which I told you earlier, and histopathological studies like brain, gill, heart, liver, intestine, all can be done. So many applications are there for this zebrafish. Even for Drosophila, but zebrafish has more applications. And coming to the after all this alternative animal models, the amount of advantages we have for alternative animal models. So recent trends and real-time evidences are there, which I'll be talking about, where all the types of tests which are done in your real-time evidences, and some articles, some evidences which I'll be sharing it to you. So, as I told when I did my first webinar in Samruddhi, artificial intelligence was one of the most hot topics, and got a very good response for that. And now also, even for toxicity studies, artificial intelligence recently research on the development of artificial intelligence based computational toxicology models that predict toxicity without the use of animal testing. Then understand the recent development of these models. We analyze databases. So all the types, deep technology, deep mind, neural technology, everything. The databases are also there in toxicity. So here you can see third point, that is 30 different toxicity endpoints using more than 20 toxicity databases. So AI, with the help of this 20 toxicity databases, where we integrated them in the artificial intelligence software, that is a supercomputer, we have got so many 30 different toxicity endpoints. So that is the era of, that is what we are in, an era of artificial intelligence. And the use of AI technology in the development of toxicity prediction models is a new concept that will aid in achieving a scientific accord and meet regulatory applications. So here you can see in this diagram so much of data. The right now what is important is our data. Data is our key. Data is money. So what? Sorry, the data what we have here right now is totally divided into so many data, and it has been put inside the database. So studies on toxicity prediction using machine learning and deep learning, that is your artificial intelligence. So they have taken 96 papers in this article, and this toxicity endpoints are checked, and the database are being put up. All the different types of database data are being fed inside the database. And then later, after doing all that, we got a molecular descriptors and model algorithms. So we got a good algorithm to know where are we having toxicity, what are the endpoints, and how can we predict a particular toxicity without using animals. So this is what the graph shows about. It is not that clear, but definitely, I hope you guys understood what exactly I am telling. And apical endpoints and mechanism endpoints are all the this is that particular chart where it tells about different toxicity studies like acute toxicity, chronic toxicity, what are the endpoints. Then it also tells about other genotoxicity, mutatoxicity, hepatotoxicity. All that has been fed, and we have got a good result also without using animals. So we got a toxicity endpoint. Now after that, we can use it for human use also. We can find out the dose, and we can try to find out with the help of artificial intelligence. And one more experiment which was done on rabbit, that is your rabbit pyrogen test, which was a very famous test before, which we tried to find out a drug is having a pyrogen activity or not. Pyrogen is nothing but it is a property of a drug which increases the body temperature. That is a pyrogen, an agent which causes fever or increase in the temperature, which hampers the hypothalamic thermostat. So scientists first discovered this in 1950s. So we wanted to, they tried to find out with the help of these rabbits. They took rabbits for this pyrogen test. Now they are using something called as horseshoe crabs. This is one of an animal where the blood of this horseshoe crabs are taken, clots are taken, and some bacterial substances which are there in the horseshoe crabs. So this spurred the development of new tests relying on sampling horseshoe crab's blood, the horseshoe crab pyrogen test. Instead of using a rabbit, they are using a horseshoe crab. And the test using the blood of horseshoe crabs, which coagulates in response to an important group of pyrogens, which replaced 90% of rabbit testing starting in the 1980s. So this is what exactly is the timeline from 1900 to 2030. You can see in the last one, that is your 2017, from 1900 to 2017, 80% less rabbits were used for this test. And between 1989 and 1995, there was one more test invented, that is monocyte activation tests, which was the in vitro test which is done. And this was validated in 2006. And later, it was accepted in 2010. And this, as I told, the horseshoe crabs, where from 2000 they started to find out the horseshoe crab bloods, where we get to know whether a drug is having a pyrogen or not. So here you can see there's a lab which has series of horseshoe crabs there on top, which is brown color, and the blue colored bottles are the blood of these horseshoe crabs. So after giving a pyrogen, this blood is being drawn from the horseshoe crabs, and then they check whether the pyrogen is present or not in the particular blood sample or the clot. So this was one of the replacement of rabbit pyrogen tests. I guess this is a very good picture which I'm showing. The scientists are trying to validate the test with at least around 15 to 20 in a row horseshoe crabs. And definitely nanoparticles. So nanoparticles are particles which are less than 100 to 1000 nanometers, and these nanoparticles are embedded in a particular formulation or particular granules where it is being injected into the animal. So that is also one of the important what do you say, drug formulation. Normally when coming to dermatological disorders, the creams, the ointments, all of them, they have these nanoparticles. The nanoparticles are embedded in small granules or small sizes, and the gel or the emulsions all are being produced with the help of these nanoparticles, especially used for dermatological disorders, where they inject these nanoparticles and then they do the autopsy, and they sacrifice the animal, and they do the autopsy and check for different parameters like complete blood count, the venous tolerance, the pyrogen test also is done, oxidative stress, the behavioral studies. All the tests can be done with the help of these nanoparticles, which is one of the recent updates. And other things like cell proliferation studies, all can be done. So that is in vitro, and this is in vivo. So in vitro also it can be done, in vivo also can be done. So this is an evidence where first one is finding synergies for the 3 Rs, that is repeated dose toxicity tests. It was one of a report from EPA Partners Forum. So they just tell that instead of using 3 Rs, we are trying to use what is the benefit of using a 4th R. So they have given a very good article on this. If you guys are free, I guess you can go through that article. And the next article is evaluation of QSAR models, that is in silico models. So all these models, that is in silico, which are computational, where the computers are used to find out the toxicity profiles of a particular thing. So QSAR is very famous, and it is the most trending animal model which is done for toxicity studies. So coming to the in silico models, where I'll be dealing with in silico things, where we deal with computers. So here we have QSAR models, and the first database which was invented was Cosmos DB, that is version 2.0, where 44,765 unique structures are checked upon, and we have 12,538. Just a second. Yeah. 44,765 unique structures are being checked, and 12,538 toxicity studies were done. And this Cosmos database, you also guys can access it. This is the link for this particular database. That is the link which is given. So this is how it looks. Cosmos. This is a website where so many chemical entities ka data is being put, and it is one of the very famous databases. And I picked it from a UK scientist who just had a brief video on YouTube. So I just found out this database, and I tried to log in, but I could not do the real time things actually, because the login credentials, something was an issue with the login. So but still I got the gist of what exactly is there in the database. So where now in this database, no, they have given this particular what do you say, the compound, that is caffeine. So they've added caffeine. So they have found out the chemical structure and the ID value, everything is given here. So after this, when you go for downloading the toxicity data, you have for caffeine, you have basically, you can see you have everything, the safety assessment, whatever studies I told you, the acute toxicity studies, carcinogenicity, clinical studies, genetic toxicity, neurotoxicity, all the tests which I just spoke right now, all the data are being put for a particular compound. You can take any compound. I'm not just telling caffeine. There are so many compounds which you can take. You can take salbutamol, you can take diabetic drugs, insulin, all the results are already given. Okay? You just have to take the database and you can put it inside the uh, machine learning and deep learning, and you can get a particular dose. If you get a proper software for it, you can do it easily. But I am not having that much knowledge of technical knowledge about that, but still I try to summarize what exactly is there in this database. So this is your QSAR modeling. So in vitro or in silico models, where you can see this extensive validation technique is done. That is QSAR, that is quantitative structure activity relationship modeling, where large chemical libraries are being accessed, and then there is virtual screening of models. So now you can see, now you can tell salbutamol. Salbutamol is your drug which is used in asthma, which is having selectivity towards beta 2. So beta 2 agonist. So it is a beta 2 agonist. So this beta 2 agonist and salbutamol, when it tries to sit on the receptor, the ligand and the protein interactions with the receptor is studied in this QSAR modeling. So we can know what exactly is the ligand which is used for a particular drug, and what exactly can it cause in the future, the toxicity, the adverse effects. All that can be studied with the help of this QSAR modeling. And then we can do with the help of this QSAR computational techniques, we can find out, we can do in vitro assays as well as in vivo assays. More towards in vitro assays, where we are dealing with more amount of in vitro assays. So here in this QSAR modeling, they have taken Cystosoma mansoni and Leishmania donovani. So these are the two types of parasites data which is taken, and virtual screening of the drugs are done, whether it is effective for eradicating these parasites or not. So all this can be done with the help of QSAR modeling. This is just one example, but there are so much of applications towards. So this is one of the recent trends where virtual screening of drugs are done. So here, conventionally, before we used to do both. No testing was done for most of the compounds, and then we used to do animal testing. But here you can see chemical and mixtures. That is a new vision where we integrate testing strategies, that is in silico and in vitro. As I told you in your this one, in vitro and in silico models. That in silico is nothing but computational models. So in silico as well as in vitro approaches are done where pathways of toxicity, modes of action, adverse outcome pathways are all given a conclusive results. So if it is a no and we don't get a proper conclusive conclusive tests or results, we can go ahead with animal studies or animal testing. But if we have got good amount of conclusive results, whether it is a yes from both the sides, to know whether it is a negative or a positive side, we can easily consider that as an endpoint, and we can easily do the tests or clinical trial phases can be done. Then it can be approved with the help of the data. The large population data can be put inside the database and try to find out what exactly is the toxicity parameter what we are checking about. So mainly to know the safety information for all compounds few requiring animal testing. So what exactly is our new vision is to reduce the animal testing experiments. So this is what is the difference between in vitro and in silico. So here you can see in silico is nothing but chips with the computational chips, and in vitro is in your outside the body, just doing some tests which are done. And also they can integrate these two also. That is a new vision where we have all these type of tests like bioinformatics, quantitative AOPs, then tissue specificity studies, extrapolation to in vivo, individual susceptibility, prediction and risk assessment. So all that can be done with the help of integration. Yeah, this one is coming to the in vitro toxicity studies. When we do that, we get a concentration called as benchmark concentration. So in this first small image, the wells, you can see the 96 titer wells, you can see some dark spots, right? So normally in this test what they have done is they have injected a chemical, and then they found out if the color is changed from purple to reddish or maroon color, it shows that there is some toxicity. So any chemical entity where they have taken a standard and a control, and then they found out these are some of the places or the doses, serial amount of doses, serial dilution is done and different doses are given. So we can check out what dose will cause the toxicity or not. And these are some of the different types of different wells where we have toxicity parameters where it is positive for the test. So that is your benchmark concentration. With the help of all this, we get a concentration called as benchmark concentration. And then we have physicochemical properties are checked, and then calculating corresponding human plasma concentration. That is very important, right? So from in vitro we need to go to human studies. So what is happening here, we are totally bypassing animal testing. And then later, if there is some inconclusive techniques, then we can go for experimental in vitro data, then use human physiology and metabolic parameters to calculate the dose, resulting in plasma concentrations. So after all this, we get an estimate of human equivalent dose, that is HED. So which was done in phase 1 study also. Phase 1A, all the drugs which are done to know the exact human equivalent dose, and microdosing studies, that is a phase 0 clinical trial. All that is being taken care of with only in vitro toxicity studies. So that is the advancement we have come. We used to use thousands of animals to get a particular human equivalent dose, but nowadays we are having in vitro toxicity assays where we can directly find out the human equivalent dose. And this is one more example, that is silostazole loaded solid lipid nanoparticles. Again, the nanoparticles are used. Here they have checked the microemulsion method is used, and one of the drugs, that is silostazole, is your drug which is used for peripheral vascular disease. It acts like an antiplatelet. So they have checked the bioavailability and safety evaluation in an animal model. This is an article from Mariam Bibi et al. And one more is your toxicity profiling is done. A compound that is Bergmansia aurea lager. These are some leaves which are used for using acute and subacute toxicity studies in rats. So that toxicity profiling is done for ethnopharmacology. And as I told, in silico modeling, what are the advantages? So accurate results, and it involves scale in the number of chemicals that can be tested quickly. We can get quick results. That is very important. It is expensive. That is the disadvantage of in silico models. But definitely down the lane, after 10 or 20 years, we might reduce the rates also for in silico testing. And another potentially important advantage of in silico testing is its potential to identify areas for rescue or repurposing of existing drugs. So, drug repurposing or reprofiling is one of the major research topic for a particular disease condition where we have already a treatment option for a particular condition, where we try to give the same drug for a proposed condition. We are giving it to some other condition to check whether it is going to work for another condition also. So in silico model will help. That is your QSAR, that is your quantitative structure activity relationship. So here you can see, right, the picture itself tells conventionally we were using animals. Instead of that, we are using in silico techniques, that is your computational methods. And these are some of the clinical trial results where we have come across in silico models, that is liver chip results. So these chips are fed into the computers and then found out the toxicity. So these are some of the drugs which are used. That is your phyloridine. There's an antiviral property, and TAK 875 molecule, and Janssen proprietary compound, that is JNJ1. So it is a colony stimulating factor receptor kinase inhibitor. So all these drugs are used for clinical trials with the help of only in silico models. So that is what I want to tell in this. I just don't want to tell more about this. But what you should know here is there is no animal testing was done for this particular drugs. Directly computational techniques were done, and then we found out what are the toxicity profiles, and then clinical trials were started. Yeah, this is also one more type of tissue engineering where we try to take different tissues like hepatocyte, then cartilages, chondrocytes, endothelium, epithelium, all the different tissues which are taken, and then it has been put in a computerized chip with some different types of techniques are there. First one is lung on chip, and the second one is liver on chip. So on chip is your hepatocyte, and lung on chip, that is your epithelium. Lung epithelium, from the lung epithelium you take a tissue and you create a chip, and you try to put some databases, and you try to find out from the computational method whether this particular test compound is having toxic potential or not. So basically, what are these in silico? These are some of the major in silico models. So what I want to tell about these recent trends are, we have tried to reduce the amount of animal testing, and it has almost come to a end only. So where only some 10% or 20% of animals are used for particular animal experiments, where we require complicated what do you say, the complicated treatment options which has to be done, or there is no new treatment for a particular disease condition. For that we need to have animal testing, but already we are having so much of data, and we are having so much of technology, so where we try to use the computers, and with the help of these chip methods, in silico methods, we can find out, especially on all your vital organs. So that is what I want to tell, basically, to know what are your alternate models other than using animals. So in silico, in vitro test, you should know. So those are the two things which you should know. And the third one is your alternate to animal models. All the three, which are your recent trends for your toxicity studies. And the database, the toxicity database, where we try to put in a lot of toxicity data. So these are some of my references which I have taken. I guess I've taken from all the articles, and I've referred some books also. And I try to come up with a presentation. I guess I was not boring, and I try to cover as much as possible. So Thank you so much. I just wanted Dr. Raghu to speak, and if there is any questions, you can come up with the chat box.
Yes, Dr. Karthik, it was a wonderful presentation. Yes, any questions you can message in the chat. It will be answered by Dr. Karthik.
I just wanted to know, how do I access the chatbot?
No, 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 it is in your webinar only.
No, I'm not able to see this chat box actually. I just wanted to please guide me for that
You can minimize your presentation.
presentation, right?
Yeah, yeah.
Okay.
Yeah, there is a chat beside. No, not there. Below,
Mm,
beside number of people, there is a chat box.
yeah,
Yes.
chat with everyone. This one. Okay. Uh, okay. So Bhagyashree Nikal J has asked about which is the best model to study aging. So yeah, as I told, aging is something where we try to tell about antioxidative property and antioxidants. So as I told, in silico models are there for aging and neurotoxicity studies. We can use zebrafish as a particular aging studies where we can get the best model out of this. And still the research is going on aging. And then about aging, what exactly is your oxidative stress is? We should know what are the parameters, like different antioxidants, what we have endogenous as well as exogenous antioxidants, which causes aging. So that has to be taken care of. And these are so many other in silico models, QSAR models are there, and that can be one of your best recent models, which can be helpful for finding to find out the model for studying aging. And next one is Dr. Vishaka Misra is asked how OECD guidelines are helpful in toxicity studies. So OECD guidelines, they just tell what should be done, that's all. But the main guidelines has to be followed is, and what tests can be done. But other than that, OECD doesn't tell much about everything about toxicity studies. But you should know what type of studies are done for a particular animal, for a particular rodent or a non-rodent. Those are the guidelines which are given for OECD. And we should always follow CCSA guidelines. IAC committee has to approve the research project, and then only we have to go with the research study. So OECD, yeah, it is one of the main bodies, governing bodies, where they give you some guidelines. But toxicity guidelines are all given by CCSA guidelines majorly, and also partly by OECD. And next one is NK999 asked that you require regulatory or IAC approval. So how can zebrafish be alternate model? Yeah, see, actually, you can see all over India, there are so many national conferences, international conferences, which are happening through zebrafish. Zebrafish has got, it has been proven in the literature also, it has good reproductive rate and it is similar to humans. And the ethical approval definitely for lower animals is not that much stringent. The guidelines are not that much stringent towards lower animals like zebrafish. But for higher animals like mouse, rats, pigs, guinea pigs, all that are having more amount of stringent guidelines to be followed. And definitely zebrafish is one of the best alternate model which can be done. And you should also have IAC approval for doing zebrafish. That is recently which has come up. It's not there in the guidelines, but I have gone to some conferences which they have told, and they have told the regulatory IAC approval should be taken. Okay, next one is Gausia Banu. Okay, that is okay. That Dr. Raghu will be telling about the recording or the PPT. And Bhagyashree Nikalje, I'm using organophosphate pesticide to induce oxidative stress. Okay. Your question is incomplete, Bhagyashree Nikalje. And Atira Ajit has asked, isn't repeated dose study different from subacute study? Yes, definitely. It is different. But the difference here is repeated dose study is a subtype of acute toxicity studies. So repeated dose toxicity studies are only done for a period of 14 to 28 days. But subacute studies are done for at least 90 days, that is 3 months. Nimasini Natarajan has asked, is it always mandatory to do in silico first and then carry out research in animal studies while using the plant product? See, that's what in silico models are having a very good predictable property, and it has having good amount of accuracy also. So doing in silico first is always better. And if you get a very good result in silico itself, but you don't have to do animal studies. And if there is any new drug entity where we don't have proper data or anything like that, definitely in silico as well as animal studies has to be done, even for a plant product as well as a chemical entity or a drug, whatever it is, any chemical. Okay, so it is mandatory. It's not like it is not mandatory, but you can do for better accuracy. Now the recent trends are telling there are so many studies which are coming for in silico models. As I told you, this QSAR, which is the most trending thing which is happening, so which integrates in vitro as well as in silico models. So where the protein ligand interactions, then the receptor molecular docking, the receptor and the ligand sitting, all that is been receptor occupancy studies, all are done, and even the toxicity studies are done. Next one is Monica P. How do we fix what dose to be used in animal experiments? That's what I'm telling you. So in the start itself, I told acute toxicity is done to fix the dose. So normally what we have is we have 1/100th of the microdosing. So there's something called as phase 0 studies, that is microdosing studies. So where phase 0 you try to find out the dose for a particular healthy volunteer or whatever. But coming to your preclinical studies, we get good amount of dose where we already know we have to do some trial and error methods. That is why acute toxicity is done where a higher dose is given and several low doses are given to find out to fix the dose. And then later we do this acute toxicity, that is FDP procedure, that is fixed dose procedure, for a period of 14 days. Okay. So can you tell about rehabilitation of animals after the study? Yeah, rehabilitation is nothing but, see, now you have taken 50 animals. Okay, 50 animals you have taken, and then from that 25 animals are dead. So that for that euthanasia and the proper care has to be taken care of, that proper burial has to be done. But for the rest 25 animals who are alive and healthy or having some pathological issues or disease, whatever, it has to be treated properly. Okay, so if it is having any disease like infection or any other immunological disorders, then hypersensitivity reactions, then some local reactions, all that has to be taken care of. That is the rehabilitation. That is the funda of rehabilitation, where we do the post experimental on animals care. This post experiment care. So that is what is rehabilitation tells about. That is the fourth R of animal research. That is very, very important, which has been added recently. Vignesh AR, the time period of zebrafish toxicity, is that as same as to rat? I don't think so, because zebrafish morphology is completely different. And compared to rat, zebrafish normally takes only 4 days to reproduce, and the reproductive rate is very high. Definitely, the toxicity studies are different from rats, but it can be some equivalent things also. You can extrapolate or interpolate so that we can get a proper what do you say, the dose for a particular this thing. And the time period you're asking, right? Time period is not at all the same. To be very sure, it is not the same at all. It is completely different. For the rat, it is more. For zebrafish, it is acute, can be even up to 1 day or 2 days or 3 days. The toxicity measures can be found out. Recently in Mysore, they conducted a workshop on zebrafish model, where we injected pentylene tetrazole, which is a convulsant, and I did the hands-on experience or the hands-on workshop. So there, where they spoke about pentylene tetrazole, which is injected, and checked the convulsant property. So we tried to give serial dilutions to the zebrafish, live zebrafish, and we found out, and cadmium chloride was added to the environment, whether it is toxic to the environment or not, and then it is checked. So that's how we did the experiment, and yeah, definitely we have got a lot of deaths of zebrafish there only. Within no time, within I guess around 15 to 30 minutes itself, zebrafish started to die because the dose was very high. So then there only we got to know LD50 was all different, and around 10 animals were given to me, to all the mates in the conference. In that, everybody's thing had at least 5 or 6 deaths. So definitely the time period is very short than a rat. Yeah, this Ames test, no, Ames test is nothing but, I'll just show you with the diagram. I just yeah. Ames test is the one which is done for mutagenicity. So here you can see, they have taken a rat liver extract, and salmonella strain is taken. So rat liver extract is put into salmonella strain, and then they have taken control and test, and they have checked whether if a compound is might be a mutagen or not. We don't know. Still, we don't know. It might be a mutagen or not. That has to be put inside that test tube, and then it has to be put in the agar, and it has to be incubated for a period of 14 days at least. And then you can see, after adding the mutagen or a particular chemical compound, the histidine rich agar, which requires basically this strain requires histidine. So this revertant number of colonies are grown here. You can see there are different light colored and yellow bright colored colonies are there. So you can see, normally either it has to be only dark yellow, or it has to be nothing. But here you can see different types of colonies here, different shades of colors of colonies, where you have revertants, that is histidine minus and histidine plus. So this shows that there are mutations which has occurred because of that mutagen. So that might be one of your potential for a carcinogen or a mutagenic property, which can be due to any genetic mutations. So that is what this Amy's test tells about. Okay, so why regulatory approval required for in vitro studies in acute toxicity studies? Why it is required, I never mentioned it requires, but in vitro studies, the regulatory approval, I don't think it is much of an importance. But acute toxicity studies in vitro you can do without regulatory approval, but you need to have IAAC, that is Indian Animal Ethics Committee approval has to be, that is your regulatory body. But still you can do, there are some relaxations in vitro studies, but in vivo definitely you should take regulatory approval. Indeed, was a graceful session. Thank you so much. Yeah, thank you so much. I guess I tried to deliver a lot of points. My main thing about doing presentations is I don't like to put more text. I like to put images and try to explain in that only. And I try to make sure you guys don't get bored, and try to make my presentation more colorful. I don't like putting more text. I like to talk and I like to interact. So thank you so much for the comments what you all have given me. What is the status of approvals of toxicity studies by CPCSCA in non-rodent big animals in Indian research institutes? Is it mandatory for all toxicity studies to do in GLP labs only? Yes, definitely. CCSCA guidelines tells, even for a rodent or a non-rodent, it has to be in GLP labs only. There is no relaxation there. Okay. And approvals of toxicity studies, yes, definitely. You need to have everything. IAC approval, CCSA. Now it is not CPCSA, it is CCSA. It has changed. Okay. If either it's for a rodent or a non-rodent, everything is same. The experiments done are all same only, and it has to be done in GLP. That is good laboratory practice, has to be taken care of. List of GLP labs in India. I guess you can Google it. You have a lot of databases actually all over India. There are so many GLP labs. In since I'm residing in Karnataka, I know so many other most of the government institutes, of the pharmacy government institutes, the medical colleges, then pharmaceutical industries, big pharmaceutical industries like No Nordisk, Micro Labs, then Novartis, all of them have GLP labs. So yeah, I guess you'll get a very good, what do you say, the record or whatever the list of GLP labs in India. I don't think I can buy had so many labs. Yeah, thank you so much, Gausia, Shivshankari, and GVA Rama Lakshmi. Okay, so okay, in silico, I just okay. You want me to just explain about in silico once again? Fine, fine. Akash, I'll just give a gist about it. Yeah, see, in silico is nothing but it is computer based. That's all. To be very simple and precise, it is computer based or computational methods which are used to find out any type of information. That is, you have something called as QSAR, that is quantitative structure activity relationship modeling. So so many ligands, the protein extracts, the protein molecules, the chemical structures, the chemical formulas, all are being put inside a computer, and then later a ligand or a particular receptor is being put inside that, and they find out whether there is molecular docking or not. If there is molecular docking or not, then we can have a better relationship. So for that, instead of using animals, we are just using computers. That's all. And they try to integrate with in vitro assays. So as I told here, in silico and in vitro approaches are done where we have integration of this, of these two. That is, see, as I told, in silico is nothing but software, data management, modeling, QSAR, PBPK, that is pharmacokinetic or other parameters, then reverse dosimetry. So all these are in silico methods, and these are some integration with in vitro and in silico. So these are some of the softwares which are done and very much helpful for toxicity studies. So that is what is about in silico. See, as I told here, with the help of in silico and in vitro, we have already got what is the human equivalent dose, which was previously calculated with the help of what do you say, the animals. Animal toxicity studies are done, and then we find out the human equivalent dose. We extrapolate it, and then we find out the HED. But here, directly with the help of in silico and in vitro toxicity assays, we have directly got human equivalent dose here only. So we don't have to do much of animal toxicity studies. So I guess I made clear about that. Who gives approval for GLP labs? Okay, OECD, the DCGA, there is NABL, sorry, what is that? NABL, that is your laboratory regulatory labs which are done, and approvals are coming from your central government. Okay. So I guess the questions are done here.
Yes.
Anybody?
Yeah, there is one more question, I guess.
Please explain rabbit pyrogen test. That's what. A rabbit pyrogen test is nothing but they inject a pyrogen inside a rabbit and a test compound. whether it has pyrogen property or not. Pyrogen is nothing but it's a substance which will cause fever, or it is going to hamper the hypothalamic thermostat. So that whether it is increasing the temperature in the animal or not is checked. So then the drugs are given. So paracetamol, which is your pyrogen, which is an antipyretic drug, which is given to rabbits to check whether the pyrogen which is given, and after giving paracetamol, whether it has antipyretic property or not. So that is the major funda of using rabbit pyrogen test. So rather than using a rabbit pyrogen test, we have horseshoe crab test, which I already showed you in the picture, where the blood is extracted and we try to give the test drug is given inside the horseshoe crab, and then later the blood is drawn out and checked with there are some analyzers where we check the pyrogens. So with the help of spectrophotometry, UV visible spectrophotometry, HPLC, all those instruments which I showed, which will have different types of pyrogens, where we try to detect the pyrogen. Okay, so that is about rabbit. It can be given in your intravenous marginal vein, then through tail vein. It can all intravenously, intramuscular, subcutaneously. Any route can be given for a rabbit pyrogen test. Why rabbits were chosen before was it had that property to induce a pyrogen. So the acetylation of COX, the hypothalamic thermostat, is weak in rabbits. So if you give a pyrogen, definitely you'll get a better result. Anta. They were using rabbits. Now we got horseshoe crab also. So that is about rabbit pyrogen test.
Okay. Thank you, guys. And everyone's question is, I think it has been answered. And everyone, many are you asking for the PPT and videos. See, I'll be making it available in our website. We are working on it. Surely we'll be given the PPT, access to the PPT, and the even the video also in the website. So it may take some time. So surely we'll provide it. Okay. And yes, Dr. Karthik, it was a wonderful session from you. Yes,
Thank you
guys,
so much. Thank you.
next class, we are planning for a clinical trial and IEC. Who all wants to attend? Can they can poll in this? Yeah, fine. Okay, we'll be planning. We'll be taking the clinical trial in the next class and even IEC webinar. And a few students they said. The Saturday, the timing that is 4 to 6, there is a problem. Do you want it on the same time or should I change it on Sunday? I have a poll for you. You can just answer this whether on Sunday or Saturday. The Saturday, whether the same time, yeah, 4 to 6 will be enough for Sunday. Okay. You can answer the poll.
Morning session, I guess it will be difficult.
No, not in the morning. We'll take in the evening only.
No, they have given in the chat actually. There's somebody, Abhinish Reddy has asked morning session, sir. But I don't think morning sessions are not easy to happen. So evening sessions are always better, even on a
Yeah,
Sunday.
yeah, correct. We are planning on the evening session only because everyone will be relaxed after the finishing the work. So yes, next class will be the clinical trial, and many are voted on Sunday. So we'll keep this next class on Sunday evening. Okay. And yeah, and once again, yeah, Sunday evening we'll see. And wonderful talk from the Dr. Karthik.
Thank you, Dr. Raghu. Thank you so
Yeah,
much for giving me this opportunity again.
yeah,
And I thoroughly enjoyed it. And the amount of response they're giving, I really liked actually.
yeah.
So,
Yes, guys, and yeah, I think most of them had many doubts. It has been cleared very nicely. And next class, this clinical trial will be more enthusiastic. Yes, and I am planning for animal. This was the animal toxicity study, and I'm planning for individual studies like animal studies in diabetes and in hyperlipidemia study and all. So we'll be planning next classes. Okay, next class will be on clinical trials. Yes, regarding the certificates, it will be provided to your mail ID by around 2 to 3 days. It will be given. And yes, and your PPTs and video will also be provided. Once the website is ready, we'll provide the link. And I think this is the end of this session.
okay, somebody has asked to talk about single nucleotide polymorphism studies.
Yeah,
Okay, we will look. Dr. Raghu, I guess he is going to plan. He's going to do it.
fine, fine, fine. We'll do. So as per your the participants' benefits we are doing. So any topics you can personally message me so that we can take up the topics and it would be helpful for you people. And I think Dr. Nivedita, Dr. Pratap Singh, and I have been five more raised hands. You can message in this chat box so that we can say the things. Yeah, surely we are organizing a individual studies on animals so that we can cover maximum of those things. Yeah.
Okay. Yeah. Thank you, Monica P.
Sure, sure. Thank you, guys. Thanks for your wonderful
Yeah.
comments.
So can we log out, Dr. Raghu?
Yeah. Okay, guys. Anything you can pin me. And thanks a lot. Thanks for your patience for 2 hours to being with us. And we'll be signing off. Thank you. Bye bye.
Yeah. Thank you so much. Bye.